Namaste students. I am Jivin Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. Today I am going to discuss about the topic tumor immunology. This topic belongs to the principles of immunology in the Department of Food and Biotechnology subjects and also in the immunology subjects. So, what is the tumor immunology? Tumor immunology is the branch which deals with the cancer and tumor and the immune system which are involved in the tumors, the antigens which are involved in the tumors. So tumor antigens response to tumor and evasion of the immune system, cancer immunotherapy and stem cell therapy are the part of the tumor immunology. In which we can discuss the tumor immune antigens response to tumor invasion and the tumor immune systems. Cancer is a stage where the cells will grow in high proportions. Cancer remains one of the leading cause of the death in the global, with approximately 20.7 million cases around the world, and they will affect all the sex very, very efficiently, and this number is expected to increase by double in the 2030. Appearance of a tumor results from abnormal proliferation of the cells, which will divide irregularly, and they will result in the loss or modification of the normal growth control. Cells which normally do not divide may restart proliferating, or cells which normally do proliferation may begin dividing in uncontrolled fashion. Means every cell start proliferating in uncontrolled fashions. Carcinogens are the agents which cause the cancers, such as the radiation like ultraviolet, sunshine X-rays, radioactive elements also induce the and damage DNA and chromosome breaks. Similarly, some chemicals also cause these cancers like smoke and stuff, and some another chemicals can also cause cancers which are known as the mutagens. They damage the DNA and which cause uncontrolled growth of the cells. Oncogenic viruses which are inserted DNA or cDNA copies of the viral oncogenes into the genome of the host target cells can also cause the uh, tumors and cancers. Hereditary certain oncogenes are heritable, means they are transferred from one generation into the another generations. If we talk about the classification of the cancer, some carcinomas, which are the epithelial origin involving the skin, mucous membrane, and epithelial cells of the glands. Sarcomas are the cancer of the connective tissues. Lymphomas are the T cell, B cell, Hodgkin's, Burkitt's lymphomas, and solid tumors. The leukemias are the disseminated tumors, maybe lymphoid, myeloid, and acute and chronic. Now we discuss about the tumor antigens, which are the part of the tumor immunology. Tumor antigens may be shown to express cell surface antigen, which are not expressed in the normal progenitor cells before the neoplasmic transformation. So tumor cells have some antigens which can be identified. These antigens have been categorized based on their nature and distribution, resulting in a complex collection of the acronym, some of which are defined as some tumor specific transplantation antigen TSTA or tumor-associated transplant antigens or TATA. First, tumor-specific transplant antigens. These are the chemical or radiation-induced tumors, each generally express a unique neoantigens which are different from other tumors induced by same or different antigens. Tumor-associated transplant antigen or TATA are the tumor-induced by the same virus express antigens shared between different tumors. And these TATA tumors consist of the membrane express virally encoded antigens and have been termed as the tumor-associated transplantation antigens. The next type is oncofetal antigens. These are the TATAs which are more or less selectively expressed on tumors, but they are also shared with some normal fetal embryonic tissues. Examples of these oncofetal antigens include the carcinoembryonic antigens and alpha fetoprotein antigens. AFE also present on the serum of the healthy infants, but decreasing by 1 year old age. These tumors stimulate the immune response. Animals can be immunized against the tumors. Immunity is transferable from immune to new animals, and these tumor-specific antibodies and cell have been detected in humans with some malignancies. And these cancer cells are different from another cells. They escape normal intracellular communications. They are very different from another cells. They allow the rapid growth. They are increased. They have increased mobility. They invade some tissues. They have mastocytosis. They evade the immune system. So these are the characteristics of the cancer cells. And immunosurveillance is a hypothesis that states that a physiological function of the immune system is to recognize and destroy malignantly transformed cells before they grow into the tumors. So immune system tries its best to kill these cells. Implies that the cells of the immune system recognize something foreign or transform tumor cells. Tumor surveillance have some specific uh, functions like macrophages and dendritic cell attack some or some have property to antigen presentation. CD8 cell-mediated cytotoxicity can also be used in tumor immunology. Antibody-dependent cell-mediated cytotoxicity have also important role in the tumor immunology. Natural killer cells have also an important role to kill these cancer cells. And these tumors can activate the immune response. Example, expression of foreign antigen with MHC1, or they can suppress the immune system also. The balance determines whether the cancer becomes clinically relevant or not. So they may activate or they may not activate. They may suppress the immune system also. So what are the basic immunosurveillance? The presence of immune cells and the tumor antigens initiate the release of the dangerous cytokines such as interferon alpha and heat stop protein. Okay. This is very important that some tumor cells and tumor antigens initiate the release of dangerous cytokines such as interferon alpha and the heat stop protein. These cause activation and maturation of the dendritic cells, such as they present tumor antigens to CD8 and CD4 cells. Subsequently, cytotoxic destruction of the tumor cells will found. Helper T cells are important. Dendritic cells are the part of this tumor immunology. And they present macrophages in dendritic cells can directly attack tumor cells, or more commonly, they can suppress exogenous antigens by the help of the CD4 cells. Similarly, some cytotoxic cells are also involved in the killing of some tumor antigens. CD8 cell T cell attach to the class 1 MHC peptide complex and they destroy cancer cells by perforating the membrane with enzymes or by triggering an apoptotic pathway. They have apoptotic pathway. So they will directly kill the cancerous cells. So this is the pathway by which they can cause destruction of the cancer cells. Sometimes some cytokines can also be used in the killing of these cancer cells. So regulating the immune system by natural killer cells, macrophages, also requires some use of the some cytokines like interferon alpha, interleukin 2, interleukin 12, which promote the natural killer cells and T cell activity and the growth factor of B cells. And interleukin 2 has important role in the T cell growth factor that will bind to a specific tripartite receptor on the T cells. And INF alpha have important role in the activation of the macrophages in the dendritic cells and the B cell and T cells. And we can also study the antibodies which are involved in the tumor immunology. Example is the direct attack by the antibody which are involved. So they can also cause the direct attack. Some antibody will block growth factor receptors and will arrest the proliferation of the tumor cells. And they may cause the apoptosis of the tumor cells. Sometimes they cause indirect attack. Major protective efforts like antibody-dependent cell-mediated cytotoxicity results in the destruction by the monocyte and the macrophages, and the complement-dependent cytotoxicity, by which some receptor will bind and initiate the complement system, and this starts with the binding of the antibody. This results in the formation of the complement cascade, resulting in the membrane attack formation and then lysis of the cancer cells. Some natural killer cells are also involved. They do not recognize tumor cells via antigen-specific cell surface receptor, but they have receptor that recognize loss of expression of the MSC class 1 molecule. So this is all about the tumor immunology. I hope the students will better understand. So the questions which can be asked here: explain tumor immunology, explain the part of the immune system which are involved, explain the cells which are involved, explain the pathways which are involved in the tumor immunology. So these can be asked. So my lecture is over here. Thank you very much.